Hi everyone, I'm Will Van Trump. I'm the Senior Director of Lab Operations at Spring Science, and I'll be talking today about AI-enabled high-content imaging, maximizing the promise and minimizing the peril. First of all, a little bit about Spring Discovery. Spring is a company that is devoted to enabling scientists to leverage the latest discoveries in machine learning to advance their different scientific programs. So what we really do is kind of fundamentally combine efforts that we've learned through drug discovery with machine learning to accelerate target identification and drug discovery in general. So a quick intro to the use of AI in high content imaging. Um, Computer vision and artificial intelligence is one of the things that has really pushed the field of imaging and particularly high content imaging forward quite a bit. And some of the earliest work really focused on things like cell pose, helping us identify segmentation and improving our segmentation through UNet type architectures. A lot of these understandings kind of bled into digital pathology and the use of computer vision classification models in digital pathology. What I'm going to be talking to you a little more today is specific to some of the work that you might do in association with high content drug screens, such as a cell painting type approach. So a real quick primer into cell paint. Cell paint is a technique that originated several years back out of the Broad, where the idea is instead of using a content-specific label in your imaging workflows, you use general morphological compartment stains, and then you use computer vision and ML to sort of extract as much information as you can out of the images that result. So here's an example of the palette that you might use. It's a very general dye to stain the nucleus, one to stain the cytoskeleton, mitochondria, cell membrane, and the Golgi. What a workflow that kind of leverages this cell paint approach might look like is data acquisition, usually through several Z-stacks, some sort of 2D projection of that Z-stack, a couple of image pre-processing steps to sort of do illumination correction, adjust for background artifacts, segmentation where you're identifying each individual cell, some kind of single cell indexing where you're either looking at those cell crops or cell segmentation images, a feature extraction step where you're extracting information about each of those individual cells. And there are a couple of different approaches here, which we'll talk about a little bit later. And then the resultant data that you get from that feature extraction feeds into some sort of normalization and alignment routine. And then once you've normalized your data, you begin to do your deeper analytics and your model generation that comes from that. Quick outline of what we'll talk about today. I'll start by discussing some of the perils and pitfalls of ML analysis, and then I'll talk about some of the things that we've learned over the years in terms of best practices, both on the analytical side and then also on the assay design and execution side for those of you that are in the lab. I'm going to take a quick pause here to sort of talk about who I intended this presentation for in terms of an audience. I really wrote this for myself, someone who has a little bit of a computational background, but is by no means a data scientist or a machine learning engineer, kind of comes out of the biological area and wants to sort of know what are the things I need to be aware of if I, in my lab, am going to collect screening data that will be fed into some of these new downstream ML-based analytics workflows. So those of you with a deep background in ML might find a little bit of this presentation reductionist, but hopefully you'll bear with me, and my hope is that it's very informative and enlightening for scientists and lab managers. All right. One of the biggest issues that we face in machine learning in general, and it is particularly pernicious in the analysis of imaging data, is the concept of overfitting. The idea with overfitting is that as you increase the complexity of the model, you actually end up paradoxically in times with worse performance. And so the idea is if you can kind of envision the number of parameters as the complexity of the model that you're building, and you look at the error value, you go through two steps. You train your model, and as your model becomes more complex, it is better able to predict the training set, and therefore you see a steady decrease in your training error as you go into more and more complex situations. However, your testing error, when you validate the model that you've generated through that training, likewise, at a very low level of parameters, a low level of complexity, you will see reduction in error, and then you achieve some sort of optimum. And then, interestingly, even as your training error continues to decline, at a certain point, your testing error begins to increase. The low end, this is what we call underfitting, which is that your model is just fundamentally incapable of capturing even the basic underlying trends within your data set. Balanced model will be able to largely represent the general effects in your data without getting caught up by the noise. And in an overtrained model, what you've effectively done is memorize the training set, and so your model becomes very responsive to individual example variability and noise and fails to capture the kind of underlying signal, which in our case would correspond to the biology of interest. Interesting, you'll see a lot of imaging software boasts the number of features that they're able to extract in their feature extraction as being this great thing that we can extract 2,000, 3,000, 10,000 features. And I think that from this, you can kind of flip it around and say, yeah, on one side, you're looking at the number of parameters that are included within your model, but it also can be the complexity of the data set that you're feeding it. As you feed more and more complex data set with more and more features, you fall prey to this overfitting regime as well. So more features doesn't necessarily result in a better model. That's one of the big things to be aware of. Another problem that is particularly insidious with our experiments in high content imaging is the idea of confounder effects. So as a little bit of a toy example, let's imagine we're training a computer vision classifier to identify images of dogs versus images of cats. So you may train the model and say, like, okay, there's dog, got it, cat, great, dog, good, dog, that's great. Okay, this is cat, got that one, great. And then we get to this example, and suddenly we have a error in our classification model where it's identifying this as dog. So if you look closely at these example image sets, you might see something in common here, and that is, it turns out dogs tend to be associated with fences to a much greater extent than cats are. And so in your imaging training set, you may have a confounder where the model is able to latch on to the presence of a fence and use that as the prediction for cat versus dog. And so it's making a call based on something that is entirely incidental to the underlying signal that you wish to capture. We see these a lot in biological data sets. For screening, you might have a compound solubility issue or an agglomeration here. This is an oligonucleotide therapeutic, which over time is agglomerating, falling out of solution, and picking up one of those morphological stains, and you're beginning to see that in the screen. The compounds of interest, in fact, also might directly influence the staining behavior. So here we have a intercalating drug that is actually competitively displacing Hurst, and so you see this sort of dose-dependent reduction in nuclear labeling. And then lastly, the compounds or conditions that you're using in your experiment, they in and of themselves may be autofluorescent, and then that becomes a very clear and obvious chemical and non-biological signal that your classifier models can latch onto. All right, moving away from confounder effects, let's talk about another big issue. When you're building a model, you want to make sure that you're feeding it a very diverse data set. So we've run into one of the examples where this can be problematic already, and that was that cat-dog example. In our training set, we didn't include a lot of images like the one that you see here, and therefore it can be seen as a low diversity training set. That would result in something like this being classified as a dog, versus if we had a much more diverse data set where we included these types of images within our training data, we would be able to correctly associate this as a cat because it's not getting tripped up by the fence. There's another area where data diversity in your training set can be really important, and that is kind of unclear edge cases. So you can imagine, what if we fed a hyena to our cats versus dogs model? If you had trained that model on a data set that was very little ambiguity, everything was clearly a cat or clearly a dog, you're much more likely to wind up with a confident prediction for what should not be a confident result in this case. Because it hasn't seen a lot of ambiguity, it's much more likely to make strong and highly confident binary calls, even in cases where it would not be entirely appropriate. if I can kind of summarize this up, when you're putting together your training data and you're collecting all the data that's going to go into training a classifier model for cell paint or other imaging types approaches, you often are faced with a trade-off. You can create a model that is very predictive and highly powerful, or you can create a model that is very generalizable. Those of you in ML might be used to this being kind of referred to as the bias variance trade-off. And what we really want to be able to do is find a way to kind of do both. We want to be able to bend this curve out so that we can kind of create a model that is both powerful and generalizable. And there are a couple of ways that we can do that. The greater that we can inject consistency into our data generation, so high quality imaging data, very consistent imaging data, very regular imaging data, that's going to help us create a very powerful model, a very predictive model, because there's not a lot of extraneous noise that is being injected into the data set. On the flip side, the way to achieve generalizability is to inject a lot of diversity into your data. And then, of course, more data sort of helps us out on both sides. But I think the key takeaway here is when you're collecting that initial data, what you want to be able to do is you want that data to have as much process consistency as you expect that you can maintain over the entire program or the entire drug screen, but no more. You want to make sure that within the training set, you are capturing the data diversity that you hope to apply the model over. So a good example is if you want to create a classifier model that is going to generalize over a number of donors, you need to make sure that your training set includes many donors. Another important part here is what I'm saying is that variability doesn't always mean noise. Variability can be meaningful in the sense that it can help guide your model towards identifying the correct biology and not getting too caught up with confounders or other effects that might pull it away from seeing the true biology. So let's talk a little bit about best practices, starting on the analytical side. One of the things that you can do is really focus on your feature extraction, what kinds of features you're extracting and how you're using them. I'll talk about a couple of different approaches. A traditional approach is to calculate a number of direct measurements from each of your images. And this might be like nuclear stain intensity, cell size, cellular area. Some of them can be very obvious like those, and some of them can be a little less obvious like a Zernecke granularity calculation. And this is the kind of feature sets that you'll get from things like Cell Profiler, and then in kind of downstream commercial offerings from Revity, Harmony, or from Moldev in their Meta Express software offerings. There are some definite advantages to doing this type of feature extraction. They tend to be interpretable, as I say sometimes. I'm not exactly sure how to interpret a Zernecke polynomial calculation of granularity, but I do understand what nuclear size means and what that might mean to the underlying biology that I'm studying. That makes them, to a degree, intelligible by a scientist. Because they're a direct calculation, they're often computationally efficient, and they can be more robust against overfitting because it tends to dissuade you from having a ridiculous number of individual features. Moving forward, there has been a new approach to extracting more informationally rich features, and this is by using convolutional neural networks, so a deep learning approach. And this is what you'll see kind of coming out of the Broad with their latest incarnation of feature extraction workflows in Deep Profiler. It's also kind of what we prefer to use in Spring. And then what you'll see coming out of a lot of companies that are doing similar computer vision type tasks like DeepCell. And what this is instead of doing a specific calculation on your crop image, you're feeding that image into a convolutional neural network, which goes through a number of different steps. And at some downstream point in that neural network, you are extracting the resultant vectors that have been calculated. And these vectors are what we refer to as embeddings. And what they represent is sort of a learned, optimally descriptive set of numbers that in theory should capture as much of the overall content of that image as possible in a greatly reduced numerical vector. These have a number of advantages, and the number one is that they result in considerably better model performance. The other nice thing about them is you can leverage transfer learning. So a lot of these convolutional neural networks have undergone extensive training against very large and very diverse imaging data sets, and rather than sort of building your own, every single time you want to create a model, you can leverage one of these pre-trained neural nets. They're unbiased in the sense that nobody's picking the measurements to make. They're sort of emergent from the kind of magic that is deep learning and that draws from these networks. That also makes them robust against noise and better able to capture nonlinear and complex patterns. Just to give you a sense and really hammer home the difference in performance that one can see from using these embedding-based features, here on the left is an example of a method of action, an MOA benchmark data set. So this is a data set of self-paint images that have been treated with compounds that have been assigned to a specific method of action. And the task here is to build a classifier that predicts that method of action from the imaging data. And you can see here we've trained a classifier using traditional self-profiler features to attain an accuracy of about 72%. It's not bad, but if we kind of leverage our unbiased embeddings approach, we can actually push that all the way to 92, 93%. All right, so the type of features that you're extracting is certainly one thing to consider. Another thing to consider is how you do your normalization. This is extremely important because there are a number of confounders and there are a number of important batch effects that can kind of throw off your analytics and your model generation. And so how you adjust for that is really important. So here's a screening data set with a number of control compounds. And what I should see is clear clustering for each of these control compounds. And what should be immediately obvious to you is that I'm not getting that. And the reason that I'm not getting that is because I have a significant plate confounder effect. So there's a plate batch effect that is far stronger in the embedding space than the effect that I'm having on each of my compounds. And so what we need to be able to do is normalize for that. So we can normalize for the plate by doing a z-score adjustment for all the wells in the plate. And what that simply does is it says, let me take every value in my embedding vector for every single well. Let me compare that to the averaged embedding vector of the entire plate. I'm going to look at the difference, and then I'm going to scale that difference by the standard deviation of that measurement across the entire plate. You can actually go one step further, and rather than sort of using the entire plate as your benchmark, if you have a number of negative controls scattered out throughout your plate, you can use those negative controls as the basis for your normalization. So when you're looking at the difference, you're not looking at the difference of the entire plate. You're looking at the difference of your test well against your negative control, and then you're scaling it by the variance that you see in your negative control. So when we do this, we get kind of the best separation amongst our control compound clusters. There are a number of other important factors to consider when looking at your analysis. Just to briefly mention them, the training approaches that you take are really important. You can do a simple train versus test segregation, or you can do a more advanced cross-validation. How you generate your model, there's a number of options out there. We do a lot of work with XGBoost, but there are certainly all kinds of random forest generators, SVMs, CNNs, decision trees, regression-based models, and it's worth kind of seeing which of those best work for the data set that you're working with. And then lastly, hyperparameter tuning can be really important. And the default packages for these models come with default hyperparameter values, which are not necessarily going to be optimized for the kind of imaging that you'll get from high content imaging. All right, let's move away now from analysis and talk a little bit about assay design and execution. So certainly one thing to consider is your sample type. This kind of sample that you're going to work with is going to have its own pitfalls, but I think the really important thing to consider is to understand what are some of the underlying confounders that you might face within each of these. And some are fairly obvious. If you're working with primary cells, you need to make sure that you're capturing the different likely to see amongst donors if you want to generate a model that can generalize to new unseen donors. Even cell lines, though, can have similar effects. You want to make sure that you're either building a model that is generalized across the number of passages that you're likely to utilize within your experiment in your screen, or you want to make sure that you're running your entire experiment and program on a very narrowly constrained number of passages. Certainly, there are similar concerns and considerations for mixed populations and cold cultures and organisms as well. One thing that people often don't think about is something as simple as plate selection. The material that you're using as the substrate for your imaging work can have a significant effect on the phenotype of the cells that you're seeing. So whether you're using glass or polystyrene or a cyclic olefin type of plate, you're likely to see differences. Here on the right is an example of that. This is monocyte from PBMCs imaged in our lab under the same conditions at the same time with the same staining palette. And on the left, we have what that looks like on a cyclic olefin copolymer plate, and on the right we have what that looks like on a granular biomagnetic plate. So it's not just the optical qualities of the plate, but actually the kind of the substrate itself matters. Moving on that, of course, the surface modifications are going to be important. Many of these plates, especially if you have adherent cell lines, are going to be what they call high bind, but what every manufacturer calls high bind means something slightly different. And then certainly there's a host of different chemical modifications that can be applied to plate surfaces. Again, you either want to standardize that and make sure that you're staying consistent to that, or you want to capture the diversity that you expect to see, that you want to be able to generalize across in your training set when you're building your initial model classifiers. And then lastly, kind of, the format of the plate bottom, of course, may have an effect on your imaging quality. Let's talk a little bit about plate trends. We talked about normalizing across plates, and that's a very regular thing to do. That goes a long way in clearing out the noise caused by different batches. But there's not a whole lot that you can do immediately about plate trends or differences between wells and plates. And it turns out these can be really important. And there's a lot of contributing factors that can go into this. Temperature gradients, gas exchange gradients, evaporation, all kinds of things. To hammer this home, looking at some of the initial cell paint data that was published coming out of the Broad and as part of the JUMP CP consortium, they did a tremendous amount of work to normalize their data as best they could, get rid of all the batch effects, the inner plate effects, and still what they have found is that both the row and the column is one of the clearest trends that they see in their UMAP projections. So this is ORF data, and there's all kinds of different genetic perturbations that are occurring within each of these data points, but you can really see that clear plate location bias within the overall data set. We've certainly seen similar things. When working with PBMCs, we can actually pick up subtle but significant differences in the cell type subpopulation. So if I look at the fraction of T cells in my PBMC, a mixed population at the edges of my plate versus the center of my plate, I do find that we typically have a slightly elevated fraction of T cells in those center wells versus those edge wells. So it's not merely that there's something kind of affecting the phenotype of the cells, but you can actually, in a mixed population, have an effect on the representation of your subpopulation. Things to be aware of: plate is always a significant confound factor. You want to try and keep your comparisons within a plate so that you can use that plate-to-plate normalization. And what that means often is it's better to have replicate plates than replicate wells within a plate, so you can really maximize the comparative efficacy of the plate design itself. It's also ideal to be able to randomize locations between replicate plates if you have possibility of doing that given your experimental setup. As we've talked about normalization, using controls. Your control layout can be really, really critical. Here's kind of an ideal world: if you could allot every other well in your entire plate to your control and checkerboard patterning it across the entire plate, that would be amazing. You'd have ample number of wells to normalize against, and you'd also probably be able to model out those plate gradients and potentially adjust for them. Now, that's probably not always going to be practical. Here's another example of a way that it can be done quite well. And this is out of the JUMP CP data set, where you have a good number of negative controls and that they're randomly associated throughout the plate. Then you have some sort of plate-to-plate comparison assay controls, which are kept in a consistent location on each plate. Here's an example of a kind of not so great plate design where we've got our control wells smashed to the side of the plate and always on the side of the plate. And this is probably not ideal. And then there's also the issue here, if you 180 this plate, you wouldn't be able to tell because it's rotationally symmetric in its control layout. A lot of this comes from the way that negative controls have historically been used as sort of just a QC step. And so usually, they're more of a yes-no thing, and you don't want to devote your precious locations, your most consistent locations on your plate to your controls. But when you're actually using those controls to scale and normalize your data, as you are likely to do with a lot of these cell paint type of approaches, they become actually the most critical wells in your entire plate and should be considered in terms of their placement accordingly. Finally, last other things to consider: wide field and confocal. Pick one. We generally find with cell paint, confocal is not entirely necessary and can actually result in additional confounders if you have alignment drift issues or things like that, or alignment of your pinholes on your confocal machines and things like that. Z-stacks versus TV projections. How many Z-stacks do you need? Probably not as many as you'd think. One of the things with all these that I would really hit on is any manipulation that you're making to your data or adjustment to your data or your data collection approach. Always keep in mind whether what you're doing is something for the human eye versus something that's going to fundamentally change the underlying data that you're collecting. The computer sees and the computer vision algorithms may be very different than what is apparent to the human eye, and it's certainly subject to substantially different biases. So key takeaways with sort of experimental design and execution: are you removing noise, or are you actually removing information that could be trained against to form a more generalizable model? Are you injecting potential confounders with any of the manipulations or the decisions that you're making? And then lastly, kind of always keep in mind that power versus generalizability trade-off. Do I have the ability to tightly constrain all my experimental parameters, in which case I might be able to generate a very highly predictive model, but it's really only going to be useful in a very narrow set of experimental parameters? Or I can create a more generalizable model, knowing that I'm likely to see drift over time in my larger data set, or I want to be able to generate a model that I can kind of continue to use for years across a much wider variability and range of sample types and treatment situations. So with that, I will go ahead and close. You can kind of get more information on all the things that I've spoken about at Spring Science's website, which is linked here. Thank you.